Today's topic is it is open sea cage fish culture and seaweed culture activities of NIOT. So before going into the deep into it, I will just tell about our NIOT. This National Institute of Ocean Technology, which is under the Ministry of Earth Sciences, which is an autonomous body of these Earth Sciences, which was established in 1993 at IIT. Later, we developed our own campus, and 2000 we shifted to Pallikarne. And then this center was developed during that time, around 1999. Various programs were organized and various projects were being operated by NIOT from the beginning onwards. One of the project was lobster fattening and transfer of technology. In that project, there was a group developed as Ocean Living Marine Resource Enhancement Program. Like that, one program started in NIOT. So basically, NIOT started as an engineering institute. Many of the ocean-related engineering projects were carried out. So this was addition during the 1999. Then our program of marine, later this program was modified as Marine Ocean Science and Technology for Islands, and also called as Marine Biotechnology Group. Under that, we started working. So NIOT has a lot of engineering group. As you see, core technology group is coastal processing and modeling, hydrographic and geographical surveys. Then offshore structures, mechanical design, assembly, integration, quality assurance, offshore handling, marine sensors. Under that only ocean science and technology programs is coming. In that, this center on as Andaman and Nicobar Center for Ocean Science and Technology was started. Later, it was renamed as Atal Center for Ocean Science and Technology for Islands, and it was inaugurated during the 2018. Then other projects like submersible, deep sea mining, desalination, gas hydrates, and many other underwater vehicle, autonomous underwater vehicles, and many other projects are being operated. And we have vessel management cell also. We have 4 ships, 2 deep ocean going, another 2 coastal research vessels in Chennai. And apart from that, we have a program also, ocean observation system. Through that, NIOT is deploying a lot of data buys all around Indian coast. From that, it is collecting atmospheric and ocean data, and it is sending to INCAIS as well as NIOT, and the data is being used for IMD for weather prediction and many other programs it is being used. Similarly, under the program of this ACOSTI, Atal Center for Ocean Science and Technology program, we have the following program we are operating for the many years. That is open sea cage culture, marine algal biotechnology, marine microbial biotechnology, island resource information system, then seawater quality monitoring along the Indian coast. In that, we do pollution monitoring in Port Blair and around Andaman Nicobar Islands. Then we have a program on potential drugs from the sea. Then we do environmental impact assessment studies all over Andaman Nicobar Islands. More than 80 EAS we have conducted, and we have air quality monitoring program. We have that lab at Rangachang, which is in collaboration with the 4th Paradigm Institute at Bangalore. We are collaborating with them. Then we have many other science and technology support program for our engineering group. Sometimes they came and demonstrated Indian, that is NIOT developed deep sea ROV, remotely operable vehicle, and they have also demonstrated current turbine, which can be used for production of electricity from using the current underwater current. And there were many other engineering programs being operated by NIOT. So now going to in this program, we are only going to concentrate on open sea cage culture, and that is algal culture, that is particularly seaweed culture. And I will tell a little bit associated activities also in that program, what we are at NIOT and as a marine biotechnology group of NIOT and ACOSTI, we are doing in islands and in some other places in mainland India, we are going to discuss. So before going to that, we just have a glance of our present global fisheries and aquaculture production. Global fish production now it has reached about 179 million tons in 2018. It is total valued about 401 billion US dollar, of which 82 million tons valued about 250 billion came from aquaculture production itself. At least about 46% of total production of fish is coming from that is aquaculture production. Overall, total 156 million tons were used for human consumption. Now, in the present world average of that per capita consumption is about fish is about 20.5 kg. So we have another 22 million tons of fish and other products, which are going to used for fish meal, then fish oil, and many other non-food related uses. Then in this scenario, China is the major fish producer, accounting for 35% of global fish production in 2018. Suppose if you are excluding China, then it is about Asia 34%. Excluding China, I'm telling, then America is 14%, Europe 10%, and Africa 7%, and Oceania 1%. Now, if you see the global presentation, global that fish production, if you see that blue left that graph, you can see that capture fisheries, that blue line, it is almost leveled off. That means whatever the maximum we can catch from the that is ocean, almost we have reached, and it is leveled off in the year 2010-11 itself. So in the total production, but it is increasing. That is because of aquaculture production. You can see the dotted line, which is actually it is a projected line for the 2030. So even though that capture fisheries is going to be leveled off, and it is already leveled off, and the only aquaculture production will take over, and it will help us to produce more fisheries. So now that open sea fish culture, cage fish culture is relevant. Why it is relevant, we can see that. Then if you see in global seafood consumption, actually, if you see that 2012, it is 51%, it is wild caught, and farmed was 49%. It will not be the same in the 2013, that consumption will mostly, that fishes will be coming from farmed, that is 62%, whereas wild caught will reduce. Accordingly, the production also will increase. Same that how much harvest is happening. If you see capture, it is 2011 data. It is about 154 million tons. In that 90 tons from 90 million tons from capture, and aquaculture is plus 63. In 2030 projected model, it is going to be almost equal, or it will be more. That capture and aquaculture production will be almost, it will be equal, or it will be more. So if you see that world annual per capita consumption of fish products As I said, 156 million tons. It is annual production is annual per capita consumption is about 20.5 kg during the 2020. It is based on the FAO statistics. So there is a requirement of production of more fishes. As I said earlier, it will come from the open sea cage culture or aquaculture production. So that per capita consumption also in the 2030 and 2050, it is going to increase due to protein requirement, more protein requirement. So that's what it is being. It is because of more protein requirement because of that product. World population increase in every year. So it is in 2030, the world population is expected to reach 8.21 billion. And therefore, an additional 27 billion tons of aquatic food is required. So this has to come from, we cannot go for more capture fisheries. So only by producing more fishes or protein from aquaculture based ventures, we can produce more protein. So world, suppose if you see compare the world cereal production, it is 2.2 billion tons, but world fish production it is about 156 million ton. I said then that fish protein is more attractive than plant protein because it has mean protein content of about 23%. It is overall percentage, overall average percentage, and it contains all the essential fatty acid, amino acids, and many other advantages are there for this one. It can be cultured in the fishes can be cultured in both land-based freshwater and agriculture pond and in the oceans. At the same time, it in ocean-based culture system, it will not go for competition with arable land or something like that. So we can in the coastal open sea cage culture, we can reduce the conflict of coastal farming also. Fishermen and other many other stakeholders, we are the stakeholders. They are using the coastal environment. This can be reduced if you go for even offshore culture. So now we will go for that Indian fisheries an overview. In that you know that Indian coastline, all these details. That India fish eating population is about 72.6 crore approximately. In that world population, world per capita, I told 20.5 kg, but still we are under that 20.5 kg. We are not taking that much. We are taking only 8.49 kg of around 8.5 kg of fishes per year. The active fishermen in India is about 1.25 million, whereas marine, there are 3,202 marine fishing villages, and there are more than 2,40,000 fishing crafts, and our export that in 2010-11 was 0.8. Now it is more, and maritime states we have around states and union territories of around 13. So the Indian marine fisheries potential is estimated about 5.31 million tons against the present production of 4.1 million tons in 2018-19. That is, it is only a provisional. That is, we are harvesting almost 78% of potential. So we have, as I said, we have a lot of coastline, exclusive economic zone. So we have a lot of other inland fisheries related potential resources like rivers and canal and floodplain. I have given the numbers in brackets, and there are about 12.4 lakh hectares of brackish water also. Apart from that, there are 12 lakh hectares of saline, alkaline, and alkaline affected areas also. So the current inland fish production is about 17 million tons against 9.58 million tons during 2018-19. So we are harnessing only 56% of inland fish production. Similarly, India is one of the largest fish production country in the world, shares 7.5% to the global production, contribute 1.24% of India's gross value added product, and 7.28% to the agriculture GVA. So this is the NIOT's fisheries and mariculture accomplishments. So I'll give the titles and we'll give details. Farming of fattening of spiny lobsters, mud crab fattening, then development of fish aggregating devices, then design and construction and empanelment of artificial reef, open sea fish culture. Fish culture of then it's that open sea cage fish culture objective is about developing suitable cages for Indian conditions. Then we have developed our already 9-meter cages. Now we are planning for 12-meter, 20-meter cages, and also submersible, fully automatic submersible cages. This we will see in the later slides. And this is the place where you can see that in North Bay we have deployed 4 cages, and this is the arrangement. The bottom figure can show that how that fish cages are arranged. That mooring configuration you can see that. Like this, we can increase at one place we can go for 10 to 20 cages, but 10 to 12 cages, 4 to 12 cages, it is optimal for one area for removal of, in case of removal of all the waste material, all these things. And this at North Bay, our Port Blair, we have deployed these cages, and this is the identity polyethylene that is cages. The framework is fully made up of identity polyethylene, and 9-meter. Can you see? Any issues? Hello?
You carry on, sir. Nothing.
Yeah, okay.
Please carry on, sir. Please carry on,
sir. Yeah. It is 9-meter dia cages. It is totally 320 meter cube area is available for our culture, for fish culture in in every cages. So we have, for this culture, we have done the fish culture at 3 sites. Olaikuda at Rameswaram, it is almost that semi-protected area. That then another one is Kottachatram at of Kavali at Andhra Pradesh, and North Bay also, North Bay in Andaman Nicobar Islands. And this is almost a protected bay, this North Bay. Whereas Tuplipalam and Kottachatram in this Andhra Pradesh, it is open sea, and this one at Olaikuda and Rameswaram, this is a semi-protected area. So we have tested our cages, these HDP cages, at different environment, protected environment, how they are performing when we are deploying and operating with fishes. So this is the details of sea conditions at selected locations, and we generally look for around 20-meter depth area. That is in North Bay, it is 20 meter. This, since it is 9-meter dia, 9-meter deep cages, even we can reduce the depth of that this one of the net. We can design accordingly. Different places we can install this one, and the wind speed should be a little bit normal. Even in the cyclonic conditions in the North Bay, our cages survived. Mostly the salinity is around 30 to 35 ppt only, since mostly in the open sea environment. So other parameters which was optimum in those areas. You see that this is Kottachatram area in Andhra Pradesh. This is partially protected, the Olaikuda and Rama, that is near Rameswaram, North Bay, and Tuplipalam. This is Tuplipalam, around 8 cages deployed, and all are 9 meter, whereas North Bay 4 cages, and Kottachitram 2 cages, Olaikuda, and another 4 cages deployed. You can see the performance from there. There are 2 types of culture we followed. That is, one is capture-based culture. In that method, we will collect the seeds from the natural environment, particularly from Mandavam area. You can collect a lot of this chanas, that is milkfish, and other fishes. Some there are people they are able to collect these fishes, and you can culture these fishes in the in our this cages, 9 meter dia cages. In that that is called capture-based cage culture. In that we have introduced, we have collected that this one chanas milkfish from that local areas around the Mandapam, and we introduced them into the cages, and we got very good results. And the milkfish seeds were only about 5 to 8 gram in size, which were grown up to 770 gram within the period of around 260 days. And the total is a total of around 3.5 tons of milkfish was harvested in that this one. This was our farmer secretary Sailesh Nayak, Minister for Science, came and introduced that fishes into the fish seeds into the cages. Later, you can see that good harvest of this milkfish, and even that other rabbit fish and parrot fish. Then nursery rearing of this Asian sea bass in open sea cages. You can see that bigger, that 9 meter cages. Within that, we have developed small, that is 2 meter dia cages, smaller cages. This will have mesh size of very small size, that is that will be around 2 mm dia. That is that mesh size will be very small, so that we can introduce very small fishes of 2 gram size. That will be, length will be around 2.5 centimeter, 2 to 2.5 centimeter. That means it is in very younger stage of So you can introduce, you can keep at least 3 to 4, 5, maximum 5 in each cages, these smaller dia cages. You can introduce that small fingerlings, and then you can grow up to some 30 gram size. Once it is growing to 30 gram size, you can remove this and release directly into this bigger cage, so that they will grow there. There is no need of that is nursery rearing separately in the hatchery, or you don't need a separate land-based that is land-based system for nursery rearing. Up to that larval rearing, and up to a particular size of fingerling or broodstock development, all these things you require hatchery, breeding and all. Then after that, we can use this technology for rearing up to fingerling stage. Then you can introduce directly into that. So you can see that nursery rearing of other that is COBIA fishes and other fishes. This is in North Bay, and we have done the same thing. In Kottachitram, same way we introduced Asian sea bass, and we could able to culture up to 1.5 tons of Asian sea bass. Average weight was 300 gram each fish. Then in Olaikuda, if you see that in 1 year it was cultured, and then the harvest was about 6.5 tons. That farm gate price we got was 325 rupees per kg. So there were 3 different type of fishes were there in the culture. That is milkfish, pampanos, and cobia. And each one got a different farm gate price, and the growth rate was about milkfish was 700 gram, the pampanos 350 gram, whereas COBIA around 4,000 gram. That is 4 kgs within that is 8 months after 8 months or up to 12 months. We could able to achieve this. COBIA was introduced later, and within 8 months time, it has grown up to some up to 50 gram. From 50 gram size to it has grown up to 4 kg. So it is highly profitable culture if we do COBIA culture. This is again how we introduced that sea bass in Andaman Nicobar Islands. This is 2-meter dia cages, and we brought this 10,000 seeds from SIBA, Central Institute of Brackish Water Aquaculture, Chennai. We aerated them, and within 6 hours it reached there. Then we acclimatized in this natural environment itself. Then we cut open that bags and removed the bags. Then it directly released into the small this one. You can see that 2.5 to 3 centimeter size fries we brought to Port Blair. Then it was grown up to average of around 700 to 800 gram. Some fishes even grown up to 1.2 to 1.5 kgs. We could able to. So different growth rate was there. So the problem was in this sea bass culture is they will different growth rate will be there, cannibalism will be there. Therefore, you have to segregate the fishes in different cages according to the growth. Then you have to introduce. Once they grow up to a particular size, the cannibalism will grow, and we have used artificial feed as well as pelletized floating feed and slow sinking feed, and also we used that trash fish also. So whatever the culture we have harvested, that is which was given to fishermen of Panicat area, fisherwoman of Panicat area, different beneficiaries. We have given freely that all these things. For that, that Pradhan and particularly Andaman Secretary, Fisheries Secretary came and distributed this one. Along with that, we have also given training to the people. Along with that culture time, we have given few people training also. This the entire operations. You can see other open sea cage culture. This is all. This is culture based. Culture based means you have to get that seeds from the hatchery. Another one I said the seeds you have to collect from the open that natural environment. That is capture based open sea cage culture. This is culture based. That is you can collect the seeds from the marine finfish hatcheries, and you can see that sea bass we have cultured in different times. And in one time we got 9.5 gram fry, which has grown up to 450 gram in 170 days, and harvested about 0.5 tons. It was sold to around 50,000 rupees, and the farm gate price was 100 rupees per kg. Then another time we have cultured 11 gram that seed to 478 gram in 190 days, and farm grade price was a little higher, 125. And another time we have in Kottachatram we have cultured the sea bass up from 5.8 gram to 370 grams in 180 days. So that 1.6 tons were harvested, total valued up to 1.52 lakhs, and the price it was slightly less, 95 rupees per kg that time. And then again in Andaman Nicobar Islands we didn't sell, but we got around 0.22 gram, very less than 1 gram, not even 0.5 gram fry, size fry. Very small fry size we brought, and we have grown up to that that is 700 to 800 gram. So as I said, a different growth rate was there, and so sea bass was giving a very good result. as well as like that other fishes. Then cage culture of COBIA, you can see that Brachycentrum canadum, it is we cultured successfully in Olaikuda, and the growth rate was around 15.4 gram per day in that during that particular culture time. It was 4 kg was achieved in 8 months. So initial stocking size was only 30 gram, whereas that farm gate price was 325 gram, 25 per kg, rupees per kg we got, and it was sold around 9.75 lakhs, and it was cultured around 12 months period. And this fishermen from the Olaikuda, the community, involved in this culture, and they actually largely benefited from this COBIA culture. And another time that 24 gram to 3 kg we cultured in 185 days, and 7 tons we harvested. It was sold to about 22.75 lakhs. There also that farm gate price was 325 rupees per kg. This was also done in that Rameswaram, Olaikuda. So you can imagine 22.75 lakhs if they are getting. Then I will show you the economics also. Then COBIA culture in Andhra Pradesh also we have demonstrated, and the average growth rate attained was 2.37 kg. That is in 10 months. That growth rate was when compared to Olaikuda area, it was 15 gram per day. Here it is almost half, that 7.26 gram, owing to that environment. But still it was profitable, and the harvest was around 3 tons, 3.6 tons. Then total valued about 9.95 lakhs, or almost 10 lakhs. So for that also introduction, our at that time minister came, Chaudhary Sahab came and introduced that seeds and other things, and they got very good results also. If you see the economics of fish cultured in COBIA fish cultured in Olaikuda, Rameswaram, you can see that what is the seed cost, and the 35 rupees per seed. 3,000 numbers we got, and transportation from Kodiyur, that is RGC hatchery to Rameswaram, then driver salary and that is diver salary, all these things together, we have spent, we got around 22 lakhs. In that, total expenses for the production is about, that is per kg, it is 230 rupees, 33 rupees, that is 16 lakhs. So from this one harvest, 6.4 lakhs, 46 lakhs was the net profit. It is actually, you see that this is from 1 or 2 cages. If you put this one in the similar equal numbers, if you are putting in 6 cages or 8, 10 cages, imagine that number. You can reduce the manpower cost and many other costs, and production, and that profit will automatically go up. So these are all the demonstration of marine finfish culture in different places, and our experience. And milkfish, it was giving good disease resistance. It was good, excellent, and the parrotfish also good, and COBIA also good, and sea bass, there was some issues. Pampano, we are not getting that much growth rate, but it was slow growing, but still we got a very good price for that Pampano also. So various fishes were cultured in our cages and demonstrated in different places. So suppose if you want to produce 1 million metric tons of fishes through aquaculture in India, at least we need hatchery capacity of 3,500 million tons seeds we need to produce from many number of hatcheries. So we need so for this production we need at least so many cages. many cages, and so that it can grow and we can produce. But to achieve this one, we have to establish every states we have to have our own. That is, each state should have hatcheries and cage culture system. So for in this regard, we have done some that geospatial mapping based on 15 parameters, and all around India we have studied and we created this map, and we found out some of the areas, many areas in EEZ, that is particularly in the coastal environment, including Andaman Nicobar Islands, are suitable. Some areas are unsuitable, and some areas least suitable. Like that, we have categorized, and in Tamil Nadu and many places, we have lot of in Andaman Nicobar Islands also. There are lot of places suitable for cage culture, putting our cages and with safe conditions. And based on that, we have found out in Gujarat, we have very good amount of area, and Tamil Nadu as well as Kerala also, we have a lot of areas we can do. Andhra Pradesh also, we can do a lot of Andaman Nicobar Islands. Lot of areas we have for installation of our cages. So these are all the accomplishment. We have installed cages in different places in Andaman Nicobar Islands, Tamil Nadu and Andhra Pradesh, and different species we cultured. The lowest grow, that is seed introduced was 0.2 gram, which was also grown up to 700 gram. Like that, we have developed our own technology of nursery rearing of within the open sea cage itself. These are all our accomplishments. Now we are planning for this COBIA broodstock development in Andaman Nicobar Islands. For that, we are trying to collect this COBIA. It is also available, and in Andaman Nicobar Islands, many times we have collected, and now we are planning to collect more number of COBIA fishes. And this is our another development, that is our submersible cage development. This one, which will actually which will go to deep, that it will float in the surface whenever in the normal conditions are there in the open sea conditions. Suppose if the conditions are not suitable for culture, it can go to the deep at 10 meter or 15 meter. So if the cyclonic conditions passes away, then it can come up. Like that, we are developing some technologies now. This one we demonstrate, check that North Bay. This cage can go up to 10 meter depth, and it, through hydraulic pressures, it can go down. And when we change the hydraulic pressure, then it can come up to top surface. And for feeding, you can do automatic feeding when they are inside the sea, as well as when they are floating in the sea. We can do. Now we are going for globular structures of cages. This is pear-shaped, whereas now we are trying for globular structure of cages. These are all prototype cages we tested here. And now, based on this, in Andaman Nicobar Islands, the biggest problem is getting seeds for open sea cage culture. Therefore, we are trying to develop this open sea, that hatchery at our own campus itself. We have got the designs, and soon we will be initiating construction of this hatchery. Already 11 crore is transferred to CPWD, and they are in that work. And we are also associating with the appointing consultant, all the people, and soon that work will start swing. So within this work is expected to finish within 2 years. Then we have to make for the production of this marine fin fishes. Then we have done a lot of lobster patterning in different places in Tamil Nadu, and there also we people got benefited. Then we have done a lot of mudcraft patterning program in Andaman Nicobar Islands, and we demonstrated this bamboo based and other that FRP based and iron based, iron grill based that is cages in many places. And we also introduced artificial reefs in different places, Odisha coast, and recently at Tamil Nadu coast also we have deployed this artificial reefs for helping the improving the fisheries in particular area. Also, these are all the different modules we prepared and deployed. And this is another fish aggregating devices in 10 places of Andaman Nicobar Islands we deployed. So that was also helping for the people to collect. Now we will talk about seaweed biotechnology and culture. And you know that they are all primitive plants. They are not true plants, they are floating. There are about 10,300 species world over. In that, 221 are economically India, that is important. About 145 to 110 species are used for food and phycocolloids. In India, about we have around 1,153 species, out of which 282 species reported to occur in Tamil Nadu. In Andaman Nicobar Islands also, recent report shows that 244 species of reported by Dr. Professor Morgan from Pondicherry University. We have surveyed and we have found out about 60 species from South Andaman. We are working on different products of biostimulant and alginate production and other products also we are working on. It can be used in different processes like biofood, biofuel, and biogas and biofertilizer, and particularly in cosmetics. We have collected more than 60 species. We isolate. We have preserved for DNA isolation, and this global status, if you see, Asia Pacific region is the major region producing this particular seaweeds mostly. So particularly China is the major producer, and then comes that other countries. So it is, if you see the seaweed production, nori is the popular this one, which is produced in Japan and particularly in China. The value is very much high. In India, we have culture in that seaweeds in many of the coast, but culture is actually happening only in that is Tamil Nadu and Gujarat coast. Andaman Nicobar Islands, recently we have started. India produces around 110 to 132 tons of dry weight agar. In that, we are also utilizing 880 to 100 tons of dry agrophytes for our production of various products. So in India, algin production is 360 to 540 tons, from 360 to 5,400 tons of dry alginophytes. Mostly from this agar is mostly extracted from Geldiella acerosa and Gracilaria adulis. And CSMCRI first introduced this marine algal culture at Mandapam. Then it was PepsiCo also involved in that project, and successfully they demonstrated. And now CSMCRI is also working in Andaman Nicobar Islands. They particularly worked on various species like Euchemia, then Kappa ficus, Gracilaria adulis, and the Gracilaria dura, and many other species. They introduced so many species that is for culture. These are all the seaweeds we have recorded from South Andaman Islands. We have collected from Barmanala and many places around South Andaman, and we did so many culture techniques, raft culture and open that is rope culture also. In this, we found out that pouch culture, if you put it pouch or tube net method, you can culture this seaweeds. This is from Chiliatapur. Still the culture is going on. there are it is giving good results that grassal area ideally we are continuously cultured. Then we recently also we have harvested about 40 kg of seaweeds from few rafts, and you can see this recent another 8 to 10 rafts now being in operation. And we soon will be conducting some training for this seaweed culture for the local people with the only native species, particularly Gracilaria adulis. Then we also develop various nutraceutical products like fucoidin, and we have developed a biostimulant. We have tested with our different plants, windy plants, which was developed from sargassum, different species of sargassum, and it gives good result for the seeding and that is fruiting and the growth of that plants, and it has a lot of growth factors and micronutrients, so it helps. So we have also developed that bioplastic from this that is edible seeds, which can be used for making coffee bags. So it will dissolve in the hot water, and you can drink as such, so that you don't need to throw any of the tea bags or something like that. Similarly, we have demonstrated alginate production from the local seaweeds. Like that, we are continuously doing that seaweed culture. Indian government also a lot of initiatives they are doing. For that also, CSM-CIARI, CMFRI, CIBA, NIOT, and NIO, they are involved. In Andaman also, CSM-CIARI, NIOT, and CARI also working in this aspects. With this, I will conclude that open sea cage culture and our seaweed culture, it will give good, that is fortune for particularly these Andaman Nicobar Islands. There are a lot of areas which is suitable for open sea cage culture as well as seaweed culture. So we can explore and we can involve so many agencies, as well as we can transfer these technologies to benefit of the island communities. And with that, I have a small video that I will play. With that, I will conclude my talk. This is our activities of that is our general activities in Andaman Nicobar Islands and other places. This is open sea cage installations and seed acclimatization and deploying into nursery cages, then feeding of COBIA. This is feeding during feeding time. This is particularly sea bass. This is the harvest. This is the harvest we have done up to a boat level. It is filled. Then this is fish aggregating device deployment. This is buoyancy testing at our acoustic test facility. It is about 6 meter length, this FADs. This is deployment at Andaman Nicobar Islands. You can see it is opposite to Chiriatapu. It was very long, 10 years back. These are all the islands we have installed. We have installed the same thing in the Lakshadweep, and still they are operational and they are regularly maintained. This is a artificial reef deployment at Orissa. You can see how they are deploying this that artificial reef. It is deployed at 12 to 15 meter depth. You can see they simply go and topple that this one, And after the deployment, you can see 1 year of deployment, you can see the artificial reef structures, how much bigger that so many fishes are living inside, particularly these groupers are living inside. And it gives very good opportunity for the fishermen to go and capture the fishes. It will also arrest that particularly. This is marine algal facility. We have done a lot of algal culture work. This is tubular photobioreactor. With this, we have produced a lot of compounds, particularly lutein, which is very high value product. And we have developed and transferred these technologies recently to industries through NRDC. And we have deep sea microbial culture facility. This is nowhere available in India. In NIOT has this one developed this system. You can culture the microbes collected from 6,000 meter depth in the same conditions, under the same pressure, under the same temperature conditions. You can culture, then you can bring to shore conditions, and then you can collect a lot of bioactive compounds from that deep sea bacteria and many other organisms. This has been demonstrated, and we recently done a lot of work on these areas, and we have developed some products also from deep sea marine bacteria. Then we have also ballast water testing facility. We are developing at Nellore sites. We have a lot of equipments, and in Andaman Nicobar Islands as well as our Chennai, we have many other equipments and supporting infrastructures. With that, we have developed many technologies, and we have patented a few of them. We have transferred these technologies also to the industries, and we are handholding them. With this, I am concluding my talk. Thank you very much for giving me this opportunity.
Thank you very much, Dr. Vinithkumar sir.
Yes.
For a very elaborate and very knowledgeable lecture, which you have shared with us. Now it's open for the discussion. If anybody wants to ask any query, they may please post it in the chat box, or they may ask that to the expert.
May I ask one question, sir?
Yeah.
Sir, Vinith sir.
Yes.
Sir, how far this cage culture has been taken up by the target beneficiaries, sir?
In actually in Rameswaram area, they are now started doing, sir. In some places in Kottachhatram also, everywhere wherever we are facing the problem of seed availability. Seed recently during this COVID pandemic, some of the hatcheries could not operate. Many of the brood stock died, and many problem came. Now again they started doing the private hatcheries, and even government hatcheries started doing that culture, but still COBIA we are not getting. Other sea bass and other seeds started producing in large numbers, and particularly in the coastal environment, many people are interested. In Andaman Nicobar Islands, we have already deployed 4 cages, and we brought about 8 cages, 8 cage material. We can deploy soon in Chiliatap area. We, in collaboration with Fisheries department, we want to help the fishermen or a new entrepreneur to grow the fishes. For that, administration can provide that seed and operational cost, and we will provide that that infrastructure of cage and net and all this thing, and we will be with them for culturing these fishes till harvest. One for each beneficiary, we will give one cycle or something like that, so that it is all free. Like that we are planning, but how far it will materialize, we do not know. Later, they can do their own culture. Otherwise, if somebody is interested to culture using our that cages, we deploy and maintain in Northway, and we are going to deploy at Chennai also. They can use that this one by purchasing seeds from their own resources, deploying them in our cages. They can harvest, and we will give the technology for that. They can harvest and sell it, and for that some minimum 10% or something like that operational cost will. Rest of them they can take like that. Like that we are ready to provide. So totally we have 12 kg, 9-meter cage right now at Andaman Nicobar Islands, and installed 4 already. It is being installed and it is operational for 11 years. From 2011 onwards, it is operational around 10 years. It is there in this one. Presently, we have removed all the nets from the North Andaman cages, North Bay cages, because for maintenance purpose, we are drying soon. This month itself, we will start again fish culture there with the locally collected fishes. We have collected even the carangids and cultured. It gives good results. Even fishermen can collect these small cages. They can deploy, and they can collect their fishes till it grows, and till it grows to a particular size. When it grows with minimum feeding, and when it grows to a particular size, they can sell it at a good cost like that. Because of this reason only, we want to construct a hatchery here. We, as I said earlier, in our campus, we have around 20 hectares of land. That 2 hectares, 2.5 to 3 hectares, we are using it for our accommodation, our buildings and all. Other 17 hectares of pond area we have, that we want to develop pond as well as hatchery. This one. Previously, it was that swim culture was happening here, and it was demonstrated successfully. Then that land was given to us. Now we are going to develop again. So this is the situation. And in Mandapam, many other places, people are ready and they are taking this one. This one seriously.
Thank you, sir. Thank you.
Thank you, sir.
Thanks a lot, sir, for your elaborate explanation.